Okay, hello. Yeah, my name is Leo Smit. I'm CEO of Hy2Care, and I'd like to explain to you a new breakthrough in cartilage repair, which really finds its origin in a good friend and co-founder of the company, Sanne Both. Before she was in her early 20s, she had already received 3 different cartilage repair surgeries. And nowadays, she's in pain again, and she knows that one day she needs a total joint replacement. And that's kind of the life story for millions of people. There are 2.5 million cartilage repair surgeries every year. A great deal of those people go back to their physician for a second, a third surgery, but gradually develop chronic osteoarthritis, suffering years of pain, disability, and causing a lot of costs for society as well. The reason that takes place is really originating from the current standards of care. On the right-hand side, the blue pieces, you see the standard of care, standard technologies that are being applied. That's debridement and microfracturing. The issue with those 2 treatments is that they create the wrong type of tissue. It's fibrous tissue, scar tissue, which has different properties than the elastic cartilage, and therefore it fails over a period of a few years. We are actually able to do better, much better. When you look at cell implantation, that has long-time survival data of 10 to 15 years and even longer. That works in an excellent way, but with very high prices, it is not affordable for a lot of people, and in many countries not even fully reimbursed. So what we need actually is the quality of cell implantation, but at a much more affordable cost. And that is where our CartRevive hydrogel implant comes into play. We have an implant that is made from natural materials, natural polymers, polysaccharides that biodegrade, but are also natively already occurring inside your body. So they're fully biocompatible, accepted by your body. And in this animation, you see how we foresee then the future of cartilage repair. That if you have hurt your knee, or in the future other joints, and the imaging shows that there's something wrong with your cartilage, your physician can go in with his or her scope, look at the defect, prepare it where necessary, and we will come in with our dual chamber insertion device. We will mix the 2 polysaccharides inside the device, and in a low viscous, well-flowing liquid, it is inserted inside the defect. It's low viscous, so it perfectly aligns with the contours of the defect. And then within 1 minute, our magic happens. The 2 polymers start to cross-link, and they shape up into a solid block of exactly the same shape as the defect. And moreover, the cross-linking also takes place with the surrounding tissue. So this block actually glues itself to your body. It has an open, spongy structure. It's biomimemic material, so the cells easily come into this hydrogel. And once inside, the cells multiply and start creating new personalized tissue in a timescale that coincides with the degradation of our gel. And then, within a certain period of time, you have a restored and fully functioning knee back again. Now that's an animation, and we've learned from Disney that in a computer animation everything works. So let's look at real life. Here you see the results of our preclinical equine studies. Arthroscopic surgery, the gel perfectly follows the contours of the defect. It has an open structure, and within 2 weeks, we see movement of chondrocytes towards the gel. After 3 months, our gel is gone, and a nice smooth layer of tissue has been formed. And that contrasts sharply with the chaotic mess that the microfracturing, the standard of care, used to give. If we then analyze the tissue at a 7-month timescale, we see with the ICRS-2 score that we have a 75% score, which is way much higher than the standard of care, so much better, much more natural tissue, and it actually coincides with results that are based on cell technology. So we actually do create a similar tissue as cell technology, but with a much easier method. Moving to humans, we've already completed the 10-person first-in-human trial. You see here pre-operatively the defect, the white spot on the left side, and on the right side, after 12 months, it's been replaced by a smooth layer of new cartilage. When we then ask the patients about their personal experiences, that's measured through the so-called KOOS score, which is an aggregate score defining less pain, more mobility. You see that we reached 24 points of a jump after 12 months, which is 2.5 times the target for minimal clinical relevance. And again, if we compare those data to, first of all, standard of care, we're beating them with 60 to 70%. And actually, the numbers after 12 months are similar to cell-based technologies, but then at a fraction of the cost. So putting all together, we have a new technology that satisfies basically all the unmet needs of the various actors in the value chain. It's very easy to use with no chance of errors for surgeons, creates very good tissue for the patients with a lifelong or at least more than 10 to 15 years benefit, and it's very, very affordable. That's not just my opinion. Here you see the expert opinions from the field of science, clinics, but also very noteworthy from patient organizations. The Dutch Arthritis Society is actually both a supporter but also financial sponsor of our company. Within the team, we still have Sanne Both as founder, but also Professor Marcel Karperien inside as our Chief Science Officer. And these scientists have been complemented with people purely from the industry in the strategic management team. We've had a very nice, diverse team, as you can see, with all different backgrounds, genders, and ages. And with that team, we've been able to score a significant number of milestones in the past 2 years. Notably, we are 1 patient short, almost finished our European clinical trial. So within the next few weeks, we can start the final follow-up period, and then we'll be applying for CE marking in 2026. And also noteworthy, an important milestone is the breakthrough device designation that we received from the FDA, which marks us as a therapy that first of all focuses on a very important unmet need, and secondly shows a lot of breakthrough promises. So that helps us a lot in our core project, getting market access to the US. We're currently scoping the trial and are just about at the point where we will submit our IDE dossier. We expect full clarity on that at the end of the year, early next year, then use the following year for further preparations, and then intend to treat the first US clinical patient in early 2026. And that will then coincide, more or less, with receiving the CE mark and starting to treat commercial patients in the European Union as well. In order to get to those 2 patients, those 2 milestones, we are currently fundraising. We're looking for €9 million or $10 million to bridge that period. We have commitments from the European Accelerator. We have commitments from our own shareholders. We are looking for 1 additional lead investor with a healthcare background to complete this round. And should then, for that investor, the appetite be a little bit bigger, then be aware that after this round, we will have another 30 million round in order to complete the full American trial. So, summing up our presentation, I hear it a lot, and for many, many years, we've always believed that cartilage cannot heal, cannot be rebuilt. That's actually a myth, and we've busted that myth. Your body is very capable of rebuilding cartilage as long as you give it the right support, the right nutrients, the right trigger. And that is what our CartRevive hydrogel implant does. And with that, we are moving ahead towards what you could say the final frontier in orthopedics, trying to prevent the metal and the plastic to enter your body, but going towards full and effective joint preservation. So we're very enthusiastic about that journey. And if I've been able to share a little bit of that enthusiasm with you, then let's talk and see if we can make the journey together. Thank you.